Pharmacy in Brooklyn, where we teach future pharmacists. And I'm here today with Jackie Cusimano, who is an infectious disease pharmacist who teaches at our college, and our students, Ramanpreet Kaur and Liliana Armasaru, who are going to be pharmacists in about 2 weeks. So it's incredibly an exciting time for us and for them. And I'm really excited to be here. Can everybody hear me okay? Yes, I'm going to, let's see, I can't see anybody, but hopefully, yes, okay, I see some thumbs up.
Yes.
Beautiful, thank you. Okay.
Yes.
Thank you very much. Okay, so what I want to tell you is, again, I am not a medical doctor. I am not a pharmacist. I am a basic science biochemistry person. And I am not here today to convince you to get the vaccine. I'm not here to convince you to not get the vaccine. I'm here to help you learn how vaccines work, to help you separate fact from fiction, and to give you the language and the understanding you need to bring your questions and your concerns to your doctor, who knows you and your situation best, to help you make the decision that's right for you. And now we will talk a bit about the COVID-19 vaccine. But before we go into how the vaccines work, I wanted to go back and talk about what is a virus and how do viruses work? Because of course, SARS-CoV-2, which causes COVID-19, is a virus. So all a virus is a container or a shell made out of protein. And inside that container is instructions to make a new virus. And that those instructions can be made of either RNA or DNA, depending on the type of virus. So that's all a virus is. It's a container filled with instructions. A virus does not have the machinery to replicate itself on its own. It actually needs to hijack a host and use the host machinery to make more copies of itself. So that's what happens when a virus infects a person. So the virus attaches itself to the human cell. It then injects those instructions into the human cell, and the human cell reads those instructions and follows them to make more copies of the virus. Once a human cell has made many copies of the virus, the human cell breaks apart and releases those newly made viruses into the body. Each of those newly made viruses then attaches itself to a new cell, and the cycle repeats itself. So that is how viral infections work. And thank goodness, we typically have an immune system that protects us against pathogens, which is a fancy word for viruses, bacteria, anything that might cause infection. So we have two different parts of our immune system. We have something called the innate immune system and the adaptive immune system. Now, I relate the innate immune system to the security guard at the mall. So they're always on call, 99% of the time, nothing happens, but they will respond to anything and everything that goes wrong. Now, usually the security guard is kind of scooting around or hanging out, but sometimes somebody breaks in, somebody tries to shoplift, somebody steals, somebody gets attacked, something happens. So our security guard responds. And they not only respond to try to neutralize the situation, they call for backup. And in our bodies, that backup is the adaptive immune system. So we talked about how the innate immune system is the first to respond, and it is generic. It will respond to anything and everything that goes wrong. The adaptive immune system is specific. It responds to this particular pathogen, and it creates something along the lines of a wanted poster. It says, okay, this pathogen, this virus, this bacteria, this is what it looks like. Keep an eye out for something that looks like this because this is a problem. So if you see it again, respond. Kind of like a wanted poster at a police station. So now, once a particular pathogen has invaded once and our immune system has responded, our security guard is never quite fully asleep again because there's that wanted poster. And our immune system is always on the lookout for if it ever sees that particular pathogen again. So that if that particular pathogen does decide to enter again, our immune system doesn't have to wake up and identify there's a problem. It's already ready to respond. And you get an immediate response that is faster and stronger than the response the first time. So the first time you saw the pathogen, it took a while to build up a response, but you finally build up a response. The second time you see the same pathogen, your response is quicker, stronger, and more effective than it was the first time. Okay, so we talked about security guards and police officers. What does this actually look like in our bodies? Well, the innate immune system is made up of several different types of cells called neutrophils, basophils, eosinophils, dendritic cells, and NK cells, which stands for natural killer cells. And what do these cells do? These cells will attack any perceived pathogen, bacteria, virus, you name it, they will attack it. And the way they attack it is they release histamines, which are chemicals that kill pathogens. And you've probably heard of or taken antihistamines. So what that is when our body has an allergic reaction, our body is attacking pollen or peanuts or dust or something that really isn't harmful, but our body sees it as harmful. So we release these histamines, which causes the itchy eyes and the runny nose and the sneezing. So antihistamines kind of tame that response. These immune cells also engulf pathogens. They wrap around them and digest them. They eat them. So that's how our immune system protects us against viruses and bacteria. So those are the cells of the innate immune system. You also have the adaptive immune system. That's the backup. That is specific. So it responds to a specific pathogen. And the adaptive immune system is made up of T cells and B cells. T cells will seek out the cells that are infected with the pathogen, and they recruit additional neutrophils, macrophages, T cells, and B cells to respond. So they say, hey, the problem's over here, come and attack it. B cells are the cells that produce antibodies. Now, antibodies are Y-shaped proteins that will bind to a specific pathogen and label it, making it easier for immune cells to find. So antibodies don't actually destroy a pathogen, but what they do is they label it and make it easier for the other cells of the immune system to find and attack. So an analogy I like for antibodies is Where's Waldo? So what I'm showing on the screen is a busy beach scene where Waldo is in here somewhere, but it's really hard to find because there's lots of people wearing red and white striped shirts and people have red and white striped towels and there's lots of blue, and it's kind of difficult to find Waldo. But if I circle Waldo and point an arrow to him, he's a lot easier to find. And that's basically what antibodies do, is they label the pathogen, they label the virus or bacteria, making it easier for our immune cells to find and destroy. So that is how viruses work and our immune system works. And I want to just take a pause at this moment to see if there are any questions before we move on to how vaccines work.
And if anyone,
yes. Jacqueline, this is Frances speaking. I'm just wondering is it better to have high levels of these neutrophils and basophils in your blood when they take a complete blood count? Is it better to have high levels?
That is a really great question. So is it better to have high levels of neutrophils or these cells when they do a complete blood count? So I will say, Dr. Cusimano, but please chime in. But from what I know is there is kind of a normal range that you're supposed to have. And if it's too low, that means your immune system may be having a problem and is not functioning properly. If it's too high, that's an indication of infection. Because when there is an active infection, your body creates more of these cells to respond to the infection. So by testing the level of these immune cells in your blood, the doctors can know, is your immune system functioning properly? Or might you have an infection? So when they look at what's called a white cell count, these are all types of white cells. That's what they're looking for.
Great, that was very clear. Thank you very much.
Great question. Other questions? Let's see.
If anyone on the phone has a question, you just dial *6 to unmute yourself so that we can hear you. Going once, going twice.
There'll be more time for questions later, so no worries.
Great.
Okay, so that was how that's how viruses work and how immune systems work, pretty much. So now, how does a vaccine work? And what you see here is this is my colleague, Kyle Hampson. He's a pharmacist who teaches at Long Island University as well. And he's vaccinating one of his colleagues at the hospital. So how does a vaccine work? What I'm showing on the slide is a picture of a tiger. Now, a tiger is a very dangerous animal. You would not want to be in the same room as a tiger. If I have a security guard, my immune system, and I want to teach them that a tiger is a dangerous animal, I could take them to the zoo and dump them in the enclosure with a hungry tiger. They would learn very quickly that a tiger is a dangerous animal. But the problem is, by the time they're aware of this, they're lunch. So that is not a very effective way to train my security guard to avoid or keep away from tigers. Another method that will likely be more effective is to show the security guard a piece of tiger skin. Now, a piece of tiger skin by itself is harmless. It has no teeth, it has no claws, it cannot attack you. But its pattern is distinct enough that if you see this particular pattern of colors and stripes, unless somebody's making a very peculiar fashion choice, it is likely on a tiger. So if you show the immune system this piece of tiger skin, it's harmless, but the immune system doesn't know that. All it sees is something's invading, something is different, attack. So our immune system identifies this as an intruder, responds, calls for the adaptive immune system, send backup. The adaptive immune system responds, your T cells and B cells are created, antibodies against this tiger skin are created. And if you remember, our adaptive immune system also creates this metaphorical wanted poster. Hey, keep an eye out for something that looks like this. So now our immune system is keeping an eye out for the striped tiger pattern, which means that if we ever encounter an actual tiger with the teeth and the claws that is harmful, our immune system is trained and ready to respond quicker, stronger, and more effectively than it would have had it not previously been exposed to the tiger's skin, so that it knew what to look for. That is pretty much how vaccines work. So how does the COVID-19 vaccine work? So if you remember, a virus is a protein shell that contains RNA instructions to make a new virus. Now, on the shell of the COVID-19 virus, SARS-CoV-2, is a particular protein called the spike protein. And in the picture of the virus that I'm showing, it's shown by a little red squiggle. And this red squiggle, this spike protein, is on the outside of every COVID-19 virus. But this particular protein in this particular structure is not found in human bodies. So it is distinct, and it will, like the tiger skin allowed our police officer to learn to recognize the tiger, the spike protein by itself is harmless. It cannot cause infection, it cannot make you sick, it cannot do anything by itself. But it is distinct enough that if your body can learn to recognize the shape and structure of the spike protein as danger, then if you ever are exposed to the actual virus, your body will be able to respond quickly because it's seen this before. So how does the COVID-19 vaccine work? For Pfizer, Moderna, Johnson & Johnson, the vaccines contain instructions for our bodies to make this spike protein. So you inject the instructions into your arm, your body starts to follow those instructions and makes copies of the spike protein. That spike protein is not supposed to be there. So your immune system says, wait, problem, call for backup. And the adaptive immune system responds. It forms these B cells and T cells. It forms these antibodies against the spike protein. So now we effectively have a wanted poster. Our immune system has been trained and it says, hey, keep an eye out for something that looks like this. So now, if you're ever exposed to the actual virus, you can respond quickly and effectively. And one common misconception I've seen is that a vaccine does not prevent anybody from catching a virus. And that's true for any vaccine, not just COVID-19. A vaccine does not prevent you from catching it, it prevents you from getting seriously sick from it because it allows your immune system to respond more quickly and more effectively. And in many cases, your immune system is able to respond quickly and effectively enough that you can keep the viral load low enough that you're not an effective transmitter. What that means is many vaccines enable you to fight off the virus so quickly that it doesn't get high enough for you to be able to spread it to somebody else. Work is still being done to determine if that is true or not for the COVID-19 vaccine. From the data that's currently coming in, it looks very promising. It looks like it does limit how a vaccinated person, even if they catch the virus, might be less able to spread it to somebody else. But we don't know that for sure yet. Work is still being done on that. So again, a vaccine does not prevent you from catching the virus, but it does prevent you from getting seriously ill from it because your immune system is trained to respond more quickly and more effectively. So I have been vaccinated. I got the Moderna vaccine. So my body had the instructions to make the spike protein. I made the spike protein, my immune system responded, and now my immune system has been trained and is ready with antibodies so that if I ever were infected with the COVID-19 virus, my immune system would respond quickly and effectively. So when we talk about our immune system responding, we've talked about B cells and T cells and antibodies and wanted posters and police officers. But what does this actually look like in our body? What the immune response looks like and feels like is a fever. Our body heats up because viruses and bacteria don't like increased temperatures. We experience chills and aches, fatigue, headache. These are all symptoms we associate with being sick. But really, those symptoms come from our body mounting an immune response to protect us against whatever is causing the infection. Okay. Are there any questions at this point? See, I see something in the chat. How does this process work with variants or mutations? Excellent, excellent question. So you've heard a lot in the news about variants or different versions of the virus. Now, what happens is the virus may develop mutations, which is a fancy word for changes. And sometimes these changes can make the virus more infectious. It can make it last longer or, infect people more efficiently. The important thing is, do those mutations change the structure of the spike protein? So you can imagine a tiger, we've learned to recognize the tiger's skin. There might be a tiger that has two heads, so it can attack you twice and bite you twice, so it's more dangerous. But it still has the same pattern of skin, so the vaccine will still work. If there's a tiger that can run 5 times as fast, so it can catch you much more efficiently, but it still has the same pattern of tiger skin, the vaccine will still work. Only if all of a sudden your tiger now starts to look like a zebra, or your tiger skin now starts to have a leopard pattern on it, that will impact the vaccine.
So you don't have to see my face.
Well,
Someone
wave
just
your hand,
asked
wave
me.
your hand.
Hold on one second. Does somebody have a question?
Yes.
Go
Yes,
ahead, Talk to him.
I have a question.
please.
I had the Moderna vaccine at least 6 weeks ago. I had both of them. And since I've had it, my right shoulder where I had the shot hurts on and off badly.
Okay.
And I put Salonpas on and I put Blue Emu and it still hurts me.
Well, I'm sorry to hear that. And what I would encourage you to do is actually talk to your doctor, because it may be something totally unrelated to the vaccine. I don't know of any reason why the vaccine would cause long-term pain. Is it actually in the injection site or is it the shoulder joint?
It's the round part of the shoulder, the outside of the shoulder, not the joint, the skin, the outside part.
So I would encourage you to reach out to your doctor, because I honestly don't know of any connection between the vaccine and that kind of discomfort.
Okay, thank you.
No, of course. So I hope I answered the question about the variants. As of now, most of the variants that we know of, it seems that the vaccines are effective still. So the structure and the shape of the spike protein doesn't seem to be changing significantly. Having said that, we don't know what the future will bring, what future variants will be developed. So it's really important for people to get vaccinated before the virus can change further, so that the vaccine can still stay effective. So we don't want the virus to learn that we're able to recognize the spike protein and start changing that. So we don't want our tiger to all of a sudden develop zebra skin so that we don't recognize it anymore. Other questions? See, I see something. And sorry, it takes me a minute to get to the chat. Was this the first mRNA vaccine? Excellent question. I'm going to ask you to hold on to that because we will get there in a little bit. Okay, if there are other questions, please feel free to raise your hand or chat them and we will answer them as soon as we can. So there is one of the things I said I wanted to do today is to help you separate fact from fiction and help you distinguish information from misinformation. So there is a lot of rumors, misinformation, and even purposely misleading disinformation out there. So I wanted to just address some rumors that are pretty common and concerns that people have. And one of the concerns is that this virus is mRNA, or this vaccine is mRNA based. Therefore, it has genetic material in it and can impact my DNA. Or I'm worried for my daughter, can it impact her fertility? So I wanted to take a moment and address both of these very real concerns of people. So let's talk about the genetic material part of it. Now, the virus does contain genetic material, but let's talk about what genetic material is. So to talk about that, I want to use the analogy of the United States Constitution. So everybody here studied the United States Constitution in school. The United States Constitution is the instructions our country follows to run. It's the instructions for our country. Now, you did not take home the actual US Constitution to study. That's because the document is incredibly precious. It is kept in the Library of Congress in Washington, D.C., under lock and key, under UV protection, because it's really precious and special. But the information in it is important and needs to be widely disseminated. So how do you disseminate the information? You take the words, you take the information from the Constitution, and you put it into textbooks. And every student takes a textbook home. Now, I don't know if you or your friends were one of these students, but I certainly know people who at the end of the semester had a big bonfire in their backyard and burned their textbooks because they didn't want to look at it anymore. Or some people would write, Johnny loves Amy in their textbook and doodle, whatever they wanted to all over their textbook. Did our country fall apart because Johnny over here burned their textbook and destroyed the text of the Constitution in their textbook? No, because the original document is still intact and untouched in the Library of Congress. They just messed with a copy. Now, the copy is enough to give you the information that's needed, but the original document is still safe and secure in the Library of Congress. Our cells actually do something very similar to that. So DNA is our cell's constitution. It's the instructions our cells need to run. Our DNA is located in a membrane-bound compartment called the nucleus in our cells. So DNA is protected inside the nucleus, just like the Constitution is protected inside the Library of Congress. However, the ribosomes, the machines that read those instructions to make proteins, are located outside of the nucleus. So how do you get the information from the DNA inside the nucleus to the ribosomes outside the nucleus? You copy those instructions through a process called transcription into mRNA, which stands for messenger RNA. So the instructions from DNA are copied into mRNA, and it's this mRNA, which is like your textbook. Your mRNA leaves the nucleus and goes to the ribosome where those instructions are read and followed to create protein. Now, just as an aside, I did my PhD studying how proteins move in and out of the nucleus of cells. So I spent 8 years of my life studying this arrow that shows the mRNA crossing from inside the nucleus to outside the nucleus. It's a lot more interesting than it looks in this picture, but that is what I studied for a long time. So the instructions from DNA are copied into mRNA, and it's this mRNA that leaves the nucleus and goes to the ribosome where those instructions are followed and made into protein. Now, mRNA by design is transient. It does not last long. It's made, it leaves the nucleus, its instructions are read, and then it's destroyed. If you want to make that protein again, you have to make new mRNA. So mRNA does not last long. So what that means is the COVID-19 vaccine is mRNA. It's instructions to make the spike protein. So when you inject mRNA into a person, you are not changing their DNA. The original Constitution is still locked up in the Library of Congress. All you're doing is you're sending a couple of kids home with Julius Caesar or Romeo and Juliet instead of a history textbook. You're throwing a couple of different instructions into the mix. And RNA is transient, it doesn't last long. So once your body follows those instructions to make some of the spike protein, the mRNA is degraded, just like any other mRNA, and then your body doesn't make any more. So it doesn't last forever. It is relatively short-lived. Okay, so there is no plausible mechanism by which the COVID-19 vaccine can change a person's DNA. Another common concern is that the COVID-19 vaccine can impact fertility. And where that came from is there was somebody reported that the spike protein is very similar to a protein called Sequintin-1, which is found in the placenta. This is actually not true. The spike protein is not similar to this protein that's found on the placenta. So the COVID-19 vaccine should not, trigger any sort of attack on the placenta. Now, there is currently zero evidence to support this idea that the COVID-19 vaccine would impact fertility. Many people have become pregnant during the trials, and it was just fine. They are monitoring effects of young girls. Clearly the 14 or 15-year-olds currently in studies have not had children yet, but there is no plausible mechanism by which the COVID-19 vaccine would impact fertility. And the one mechanism that was proposed has been shown to be not true. Another common concern, which is legitimate, is it went so fast. And people are concerned that clearly they rushed through this. It hasn't been vetted. It hasn't been tested. How did it go so fast? So let's talk about that. It did go fast, but there are reasons for that. So I want to talk about the clinical trials pipeline, which is how new drugs or new vaccines get approved. So once a researcher has discovered a new medication that they think will be useful, they first put it through clinical trials. So the phase 1 clinical trial asks, is it safe? So you have a small number of patients, usually healthy volunteers, who agree to participate in this trial. And they start taking this new medication, usually at doses far below what would be considered therapeutic. So it's not even enough medicine to work yet. And they're very carefully monitored for unexpected side effects. And slowly they will increase the dose until they've reached a dose that they think will be effective. But if any intolerable side effects are seen, the trial stops and the drug is over. It's not going to get approved. It's not going to move forward. Now, one thing to realize is that what is considered an acceptable side effect will change depending on what the drug is meant to treat. And what I mean by that, if you or somebody you love have gone through chemotherapy, you know that chemotherapy has pretty miserable side effects. There's hair loss, there's nausea, there's a whole lot of other things that happen. And it's awful. But we are willing to accept that because the alternative is cancer. Whereas if Tylenol had the same side effects as chemotherapy, I bet most people would probably just put up with the headache for a little while rather than go through those side effects. So what is considered an acceptable side effect changes based on what the drug is treating. But all of this is looked at in phase 1. So phase 1, is it safe? If a drug is found to be safe in phase 1, it goes on to phase 2. Now you're asking, does it work? Does it actually do what you want it to do? If it's an anti-cancer drug, does it shrink the tumor? If it's a drug against Parkinson's or any other motor disease, do motor skills improve? Does it prevent you from getting sick from COVID-19? Does it work? So all patients in phase 2 get the dose that was found to be safe in phase 1, and now you're checking, does it actually do what you want it to do? If it is found to be successful in phase 2, a drug goes on to phase 3. Now you're asking, does it work better than what's already out there? And so in phase 1 and phase 2, everybody in your trial got the drug. In phase 3, half of your patients will get your drug, and half of your patients will get either a placebo or the standard of care if one exists. And now you want to know, okay, does the group getting my drug do better than the group that didn't get my drug? So this is phase 3, and this has hundreds to thousands of patients. And once a drug passes phase 3, that's the point where they can apply to the FDA or whatever governing body in another country for approval. And if they've shown that the drug is safe and works, then they can get approval from the FDA. Now the drug is out on the market. The pharmaceutical company doesn't just go, okay, I'm done. I got my drug approved, I'm done. No, they continue with what's called phase 4, which is continued monitoring after wide release. So now you don't have thousands of people, you have millions of people perhaps taking this medication. And they are constantly on the lookout for unforeseen problems, long-term effects that weren't seen before. And we'll talk about what happened with Johnson & Johnson in a little bit, but this was phase 4. So again, the clinical trials pipeline, you have phase 1, is it safe? Phase 2, does it work? Phase 3, does it work better than what's already out there, or does it work better than a placebo? And phase 4 is continued monitoring after it's been approved for public use. Now, this whole process usually takes 10 years or more. So I worked in a lab that did the preclinical work that led to the discovery and development of a new drug for spinal muscular atrophy called Spinraza, or nusinersen. And that process, from the point my lab started working on it in 1999 to the time the drug was approved by the FDA in 2016, was 17 years. And that was honestly a Cinderella story. Everything that could have gone right in that work went right. And so that was pretty fast by clinical trial standards. That took 17 years. How the heck did COVID-19 vaccine come out so fast? There are a number of reasons for that. First, researchers did not start from scratch. So there was a question earlier about, was this the first mRNA vaccine? Well, if you remember, over the past decade or so, we had other coronavirus pandemics called MERS and SARS. Now, when those pandemics were out, scientists started developing mRNA-based vaccines for those. However, those pandemics were able to be contained by public health measures. So there was no longer the funding to support research for a vaccine because it wasn't really needed at that point. So the funding dried up, the research stopped. So those vaccines never made it to market, but they existed. So when SARS-CoV-2, the COVID-19 virus, appeared, the scientists were not starting from scratch. They had these years of work from MERS and SARS to build upon. So from what I know, this was the first mRNA-based vaccine to be widely used, but it wasn't the very first mRNA vaccine to be developed. Okay, so that's one thing. Scientists were not starting from scratch. They had years of work on MERS and SARS to build upon. Second, global cooperation to a scale at which I've never seen. So I'm pretty sure almost everyone here remembers the beginning of the AIDS epidemic, the AIDS virus. And if you've ever seen the movie And the Band Played On, it's one of the most frustrating movies you'll ever watch. It's a depiction of the early days of the HIV pandemic. And it showed that Labs in America were working on this to try and understand what this virus was and how it worked and why these people were getting sick with diseases that we've never seen before. And labs in France were working on this virus to see what was going on and how this virus is spread and why are people getting sick. And the labs in France knew some things, and the labs in America knew some things. And if they had just shared information instead of competing with each other, we could have probably had more effective treatments for HIV and more effective prevention measures for HIV months to years earlier than we did. Here, we saw global cooperation and sharing of data and information like I've never seen before. We had cooperation to the point that Merck was using its factories to manufacture the Johnson & Johnson vaccine. So global cooperation, global sharing of data, really people cooperating instead of competing made this go faster and more efficiently. Also, the fact that these were mRNA-based vaccines made it a lot easier to tweak and scale up. And the way you can imagine that is most older vaccines, you're not injecting the instructions to make the spike protein, for example. You would be injecting the spike protein, which means that you need to figure out in a lab how to make many copies of the spike protein that is the right shape, the right size, the right structure, as it would be made in the body. Whereas with this mRNA-based technology, you simply inject the instructions and the body makes it for you. So that's a lot easier, actually. And a way you can think about that is, say you had to distribute or you had to give 100 people each an IKEA dresser. Well, you could buy the dressers and bring them to your house and spend several weeks putting tab A into slot B and hammering this into that and make 100 copies of the dresser and make sure you do them all exactly the same and deliver them to your friends. Or you can take the appropriate pages from the instruction manual, make 100 copies, and send each of your friends a copy and say, here's the instructions, make a dresser. It's a lot easier to distribute the instructions and to scale up the instructions than it is to scale up the protein itself. Unprecedented financial support. Governments, celebrities, everybody was throwing money at this like we've never seen. So the people doing the work were not wanting for resources. They had whatever they needed. And the final thing that really helped this go fast is the willingness of people to volunteer to participate. One of the hardest parts about clinical trials, especially in phase 1 or phase 2, the early phases, is recruiting volunteers. Because people don't want to get injected with something that hasn't been tested in humans before. But that's what's needed for these trials. Here, people were pounding down the doors to volunteer to participate in these studies. So they were able to recruit the numbers of people needed much quicker than is usually done. So it did go fast. That is absolutely true. But there's reasons for that, and no steps were skipped. It's just because of all of these factors, it did go fast. So let's talk about the trials and how they worked. So the Pfizer trial had 44,000 people enrolled. So we're talking tens of thousands of people over a wide range of racial and ethnic distributions and a wide range of ages. Moderna also had 30,000 people enrolled. Now, the Pfizer and Moderna trials were both domestic. They took place in America throughout, 30 to 40 states. The Johnson & Johnson trial also had about 44,000 people enrolled, but this was international. These people were spread out over 8 countries across 3 continents. So all of these trials had between 30 and 44,000 people enrolled. So we're talking about a large number of people that were tested. Now, how do these trials work? So for example, the Pfizer trial, they had about 22,000 people receive the Pfizer vaccine, and about 22,000 people received a placebo shot that did not contain the vaccine. They waited at least a week after the second shot, and then they started monitoring people for symptoms. Did they actually develop symptoms if they got infected? And what they found is they found 8 people who developed symptomatic COVID among the people who received the test vaccine, where 162 people developed symptomatic COVID among the people who received the placebo shot. So this is where that 95% effective number that we hear all in the news, this is where it comes from. 8 people, or 5% of people who got sick, received the Pfizer vaccine, whereas 162, or 95% of people who got sick, did not get the vaccine. So that's where that 95% effective number comes from. So what's important when you're comparing the data about the different vaccines is that they're actually each reporting something slightly different. So Pfizer is reporting that it was 95% effective at preventing disease a week out, 7 days out. Moderna had a similar report, 94%, but they started measuring 2 weeks after the vaccine. And Johnson & Johnson started measuring 4 weeks after the vaccine. But their vaccine is only 1 shot, not 2. It is stored in the fridge. It does not need the super cold storage conditions that Pfizer uses, so it's easier to distribute to certain people. So there are certain, any of the 3 are fine. Now let's talk about side effects. So these studies, side effects are expected. People have reported injection site pain, headaches, muscle aches, fevers. I wouldn't expect shoulder pain to last weeks on end, so I would definitely encourage you to talk to your doctor about that. But what I encourage you to think of is, where have we seen these side effect symptoms before? We saw those symptoms when we were talking about the immune response. This is our body actually mounting an immune response and attacking this spike protein. So these, what we call side effects, is actually what you want to see. You want to see your body responding to this perceived infection. Now, in the trials, they monitored patients for at least 8 weeks after getting the last vaccine, and they saw no significant safety concerns during the trials. And it is very unusual for unexpected side effects to appear more than 8 weeks after vaccinations. So while 8 weeks is probably a little bit shorter than most studies go before they submit approval, it's long enough for vaccines, because you're not going to see a side effect, a new side effect, start to appear more than 8 weeks after you receive a vaccine. Another thing that's been going around is people say, oh, I'm just going to get COVID and get natural immunity that way. So you're right. If you actually were to get infected, you would build natural immunity. But the risk of illness and death from getting COVID far outweighs any potential benefit of natural immunity that you might gain from that infection. So relying on actually getting sick to develop immunity is just not a safe plan. And what about kids? Why can't kids get the shot yet? So I love this picture because it's absolutely adorable. What it shows is 3 children wearing grown-up business suits that are far too big on them, and they're super cute. But what this picture is meant to show is that kids are not just little adults. So you can't just say, oh, this kid weighs half as much as a grown-up, therefore you give them half the dose of this medication. Kids metabolize medicines differently. Kids, have very different response to medication. So just because the medicine is safe and effective in adults doesn't necessarily mean it'll be safe and effective in kids. So kids need their own clinical trials. Clinical trials in kids are actually quite complicated because kids are vulnerable. Kids are a protected group. Mommy says I have to take this medicine. Okay, I guess I have to take this medicine. So kids are very protected so that you make sure that a child is not being forced to do something they don't feel comfortable doing. So because clinical trials in kids are more complicated, most companies will start trials in adults first. And then once the drug is shown to be safe, effective in adults, then they will start slowly lowering the age group they're testing in. So where are we in terms of trials with kids? As of the end of March, Pfizer was completing a phase 3 trial where they were studying about 2,000 12 to 15-year-olds. And they say that the vaccine appears to be safe in this age group with no new side effects that they hadn't seen in adults. And it was effective. You got a similar 95% effective rate that you saw in grown-ups. So they're submitting that to the FDA to get approved for ages 12 and up. And they are now mounting a global study of the Pfizer vaccine in ages 6 months and up. So hopefully soon we'll be able to start vaccinating children. Moderna is also completing a trial with 12 years and up, and they hope to release their data by summer. Now, AstraZeneca was planning to start a trial with children in February, but that trial was actually stopped because they started seeing blood clots in adults with the AstraZeneca vaccine. And this shows you that this is phase 4. Once the drug was released to the public, they started seeing this blood clot problem that was not seen in the trials. It was within 2 weeks of getting the vaccine, so it was well within that 8-week limit that they looked at. But because they were starting to see blood clots with AstraZeneca, they stopped. They stopped the trials in kids until they can understand more what was going on. Speaking of stopping, what the heck happened with Johnson & Johnson? So what happened was, after about 6.8 million people got the Johnson & Johnson vaccine in America, 6 of them, 6 premenopausal women, experienced life-threatening blood clots within 2 weeks after receiving the vaccine. It's not entirely clear if those blood clots are directly related to the vaccine, because 1 in a million, which is what this was, it was 6 women out of almost 6 women out of almost 7 million people who got the vaccine. 1 in a million is actually less than the risk of blood clots just in the average population. So it's not entirely clear if these blood clots were directly related to the vaccine, but just to be safe, they said, let's stop and check this out. So if you got the Johnson & Johnson vaccine, or does that mean it's unsafe? No. Again, this is truly a legitimately 1 in a million event. So for most people, the vaccine is safe and is effective, but they wanted to take a stop and see what was going on so they can understand, even if this is very rare, maybe this means they shouldn't give the vaccine to premenopausal women. Maybe they find that it only affects people who had a hamburger for lunch the day before. I'm making that up, but they wanted to see if they can understand what triggered these blood clots in these women. So the good news is that this does show the system works. This is phase 4. Once the drug has been released, if there aren't any problems or concerns, it's pulled, it's paused, it's stopped, and people investigate to figure out what's going on. And it is possible that these blood clots are unrelated to the vaccine. So the CDC and the FDA both reviewed the analysis. And as you heard recently, they did suggest to lift the pause and continue giving Johnson & Johnson vaccine again. So if anybody got the Johnson & Johnson vaccine, don't worry. Any blood clots always happened within 1 to 3 weeks after being vaccinated. I think the longest one was 13 days after, if I remember correctly. But if you have any symptoms of blood clots, like severe headaches, abdominal pain, difficulty breathing, or swelling in your legs, definitely contact a healthcare provider right away. And these green images are from the Johns Hopkins Bloomberg School of Public Health, and they gave me permission to use them. So I want to thank them for that. And again, we talked about that there is a 1 in a million incidence of these blood clots in people who've received the Johnson & Johnson vaccine. For context, the risk of blood clots from people who use hormonal birth control pills is approximately 1 in 2,000. So we're willing to put up with that risk for birth control pills. And I also wanted to share with you, and I will give Alana these slides so she can send them to you, reliable sources of information. There is a lot of information on the internet. Some of it is reliable and some of it is not. And it's really hard when you don't know, when Google pulls up a site, to know if this is reliable or not. So here are some reliable sources of information that have language that is meant for the public. So you have the CDC and FDA websites. There are parts of those websites that are meant for scientists and researchers, but there are other parts that are meant for the public. Your state or local health department. So I've provided the COVID-19 website from the New York state government, the National Medical Association, the World Health Organization, and Gavi, which is a worldwide organization about vaccine information. And then finally, I want to review, we did get a couple of questions submitted in advance, so I thank you for that. And so I think some of these were addressed during the talk, but we'll just revisit them now. So a person had asked, if I had the Moderna vaccine, would it be suggested if I stick to that same brand for the booster shot, or should I take the first one that is available? So those guidelines have not been released yet. And actually, before I go on, Ramanpreet and Liliana, did you guys have an answer prepared for this? I don't want to step on your toes.
Thank you, Dr. Novat. So actually, I think you're about to give the same answer that I was.
Go for it.
Yeah, so we're currently waiting for the guidelines to be released on the booster shots. I know everyone has questions about these booster shots, and because they're not they haven't completed the approval process yet, we don't have a lot of information either. But as soon as they're approved, we will have a lot more guidance on who will get which shot and when the booster is recommended, for which age groups it's recommended, and all of that information is going to come out as soon as we have those shots available. So thank you. That was a very good question.
Excellent. Thank you so much. And one thing I will add is I don't know if you saw on the news, but there was one person at least who got, I forget which order it was, but he got, let's say he got Pfizer as his first shot. And then when he went for his second shot, they realized they accidentally gave him the Moderna one. But if you think about it, the way both vaccines work is they're providing instructions to make the spike protein. So it shouldn't matter which brand of instructions you're reading. If you make the same protein, your body will respond the same way. But the guidelines have not been released yet, so we don't know what they're going to say.
Absolutely, and just adding on to that, I think in Europe, initially when the vaccines came out, because there was a shortage, they were allowing the Pfizer and the Moderna vaccine to be used interchangeably. So there is a good chance that if you have, say, the Pfizer vaccine series, and the only option was to get the Moderna booster, I think it should be okay to get it. But again, we're just going to wait on the guidance to be released. Excellent.
I will say, as a scientist, one of the most exciting answers to a question, at least for me, is we don't know yet, because that really means you're on the cutting edge of what's known, and we really are. So another question that came in was, it seems that more people are having side effects from the Moderna vaccine. Why is this? So we talked a bit during our talk today about what these side effects really are, which is an immune response. But did you two have anything more to add to that?
I believe there's a side effect that was reported with Moderna vaccine, which is not so common with Pfizer. I believe it was a rash. And I was trying to find more information about it. It's not that much available out there, but I believe that if you do have a rash, and if it's after 4 hours after getting the vaccine, you shouldn't be worried about it. If it's within 4 hours after you get the vaccine, then it's a more serious reaction. It's a more serious case, and then you would have to contact your doctor right away. But if it's 4 hours after, don't worry about it. It will go away. It may take 5 to 2 weeks, but it should go away. It's just a reaction that your body has, your immunity has, well, just like Dr. Novat said before.
Excellent. And I think it's important to say also that a rash is a typical part of an immune response. If you have an allergic reaction, one of the symptoms of that is a rash, because your body is responding and trying to attack what it perceives as a danger. So that's probably what's happening here too. But of course, if anybody has an allergic reaction to the vaccine, you definitely want to get medical attention right away. And that is why there is always medical professionals available at a vaccine site. So people get nervous because they see ambulances on standby at high school gymnasiums or wherever vaccines are being given out. The reason for that is typically you get a vaccine at a pharmacy where there are healthcare professionals around. If you're getting a vaccine at a healthcare gymnasium, there's not going to be that availability of healthcare professionals who know how to respond if there is an emergency. So that's why they have ambulances on standby, just in case there's a problem.
We usually monitor our patients. We both work in a community pharmacy, and we always have our patients have a seat and wait for 15, 20 minutes, or stick around the pharmacy for some time to make sure that they're okay and they don't experience any side effects right away, or unusual side effects.
Yeah, and just I know I was talking about the vaccination course that we go through. We're also CPR certified. And in that 9 to 20 hour course that we take, we are made aware of all the vaccine, like all the reactions they could possibly have to a vaccine, as well as how to manage those reactions.
Yeah, and we have the tools available. We have an EpiPen in case of anything. We have pharmacists, and then we have a hospital right next to a pharmacy. So we're actually, like, in case something would happen, thank goodness nothing happened with our patients so far. We've never had like anyone experience some kind of reactions right away after the vaccine. So,
Yeah, I've been very lucky. I was just talking to Dean Novat a few days ago, and we started vaccinating with Walgreens in December. So December 29th was the first clinic that we had. We went to a nursing home. And as we're vaccinating, I remember the first thing that was coming to my mind was, oh my God, I really, really hope that we don't have anyone that has a reaction, because it's just so scary with the elderly. But thankfully, everything has been good so far, and I'm knocking on wood as I speak. Everyone has been okay so far. We haven't had any allergic reactions. And just like that's from my personal experience, going from December to now, haven't seen anything.
Thank you. And then there was a question about skipping the second dose of Pfizer. So I've heard that many people are skipping the second dose of Pfizer or Moderna vaccine, and I wonder whether I should consider skipping it. Did you have an answer for that one?
So we don't recommend skipping the second dose of the Pfizer or Moderna vaccine. It is the second dose, and it's almost a good way of thinking about it is as like a booster shot, but it's so important that you get the second dose. So I know Dr. Cusimano can go a little bit more into this, but in the studies that we've seen on the Pfizer and Moderna vaccine, after the first dose, you get about 50% immunity. You get the 50% effectiveness for the vaccine. But once you get the second dose, you get the additional remainder of that immunity. So you're able to get to that 94, 95% projected effectiveness that
Exactly.
Dr. Nova was talking
So
about.
if you get just one shot and you decide that you don't want to go for the second shot, you have a 50% chance of getting the virus. And you don't want to risk that for your family, for the sake of the family and the one around.
May I ask a question, please?
Please.
Yes, I'm going for my second Moderna shot, and I'm wondering, are the side effects temporary or permanent, and how long do they last if they're temporary?
So anecdotally, most people that I know who've experienced side effects after the vaccines, it gets better within a day or two.
Oh.
So it really, but again, what I've also seen, again, anecdotally, is that older people tend to have more mild symptoms than younger people. So my brother was in bed for 2 or 3 days after the vaccine, where my 93-year-old grandmother was totally fine.
Oh, I see. Thank you very much. That was very helpful. I'm not too scared now.
No problem. Just remember, if you do feel ill, that is your body responding.
Yes. Thank you very much. Thank you.
No problem. But of course, any concerns, definitely call your doctors. That's what they're there for.
Yes. Thank you.
Of course. And then there was this question about the achy shoulder, and I'm afraid we haven't read anything or heard anything about long-term shoulder pain. It may be something unrelated to the virus. It may be that just the act of injecting something tweaked a nerve or did something, but I would definitely follow up with your own doctor about that.